Thank you. Thank you. So I toyed around with names for this. I got inspired for this talk by information about this virus that wasn't around when I was in medical school, Nipah virus. And it's another one that's has bats at the origin. I kind of hate to see this because I love bats, actually. And first thing I want to do is quickly review the Baltimore classification, at least two of the categories. Now, in the note card I gave you, it has a link to the PDF, which has notes, some of which I'll be citing as I talk. But we're going to talk about one category 4 or a virus, like coronavirus was category 4. SARS-CoV-2. This is Japanese encephalitis virus, and it relates to the subsequent discussion. And then I'm going to talk about Hendrix virus, and which I almost think I'm seeing my name there, and Nipah virus, which are category 5 or group, I should say group 5. Those are negative sense RNA linear, and they are packaged, which means that the genome has its replicative machinery kind of packed in with it, and that's critical for its existence, actually. So the first part I'm going to discuss here is Japanese encephalitis virus. And basically it's transmitted to humans from bites of infected female mosquitoes of the Culex species. Humans don't develop a sufficient viremia such that if an infected human were bitten by a mosquito, it could infect anything else. Animals like humans that do that, in this case, are called dead-end hosts. And the converse of that is amplifying hosts. And that can happen in animals in zoonotic cycles that involve pigs, water birds, and mosquitoes can transmit it to other mosquitoes. I'll tell you about that in a bit. So as humans tend to spread out and live in the habitats of, or near the habitats of animals that are losing out, they're getting more exposed to these things. And also, in this case, vertebrate hosts like domestic pigs are an issue. So it occurs generally in temperate areas of Asia, and I'll look at a map with you in a bit. There's my map, which is resing. Is it resing for you slowly as well, or just for me? Good. Okay, maybe it's my computer. This just shows the distribution of Japanese encephalitis virus across Southeast Asia. Again, a group 4 by the Baltimore classification. David Baltimore was a scientist who developed that. He's passed away now, but there will be overlap in the distribution here with our later discussion of Hendrix virus and Nipah virus, as you'll see. But this is this Japanese encephalitis virus is all inclusive, and one reason it's important is about 3 billion people are at risk for developing it. And it's one of the things that physicians, based on there's almost a culture of diagnoses to be made, or diagnoses to be made depending on what physicians see in any one place. And they often will figure they're looking at Japanese encephalitis virus. Slide 5 here shows the pattern of Japanese encephalitis virus transmission. Mosquitoes suck blood from an infected animal and then can transmit it by biting. That's pretty straightforward horizontal transmission. And that can go to dead-end hosts like horses, in this case, horses and, excuse me, humans, or amplifying hosts, particularly pigs, amplifying meaning that they get a viremia that's significant enough that a second bite can be spread the virus further. So they become an active vector in the disease. So pigs and birds are reservoir and amplification hosts. They can produce high-level viremias for a good while. Pigs can also get infection by direct contact. And bats can also be infected and carry Japanese encephalitis virus for a long time. Now, this slide, I guess I'll just go ahead and hope that it's resed for you. It shows another bit of imagery about the vectors, but the point of this one is seropositive animals like dogs as dead-end hosts can be crucial as sentinels. They can be monitoring tools. They're not amplifiers of the Japanese encephalitis virus, but by testing dogs, you can see where it's prevalent. And so I thought that was quite an interesting aspect. Some of this I'm going to be telling you about is just to kind of make some points from general science of infectious disease and epidemiology. So high seroprevalence or presence of antibodies makes animals like dogs invaluable for assessing JEV risk, JEV, Japanese encephalitis virus, risk to humans, especially in urban and residential areas. Now, my next slide is slide 7, and it's basically to emphasize these definitions that vertical transmission can occur when an infected mosquito has eggs. They can pass the virus to their gametes. And so the offspring, male or female, can be infected with the virus throughout their development. And so that's kind of an interesting thing. It's contact through their mother or their father, so to speak. And horizontal transmission is the way we mostly think of, like from the old story about malaria, and somebody with malaria or something with malaria gets bitten and something else gets nailed by a mosquito with it that's infected with that disease, and they transmit it horizontally. So horizontal versus vertical transmission. And one somewhat alarming point is that Japanese encephalitis virus can be transmitted from a mother to a fetus and can result in stillbirth. And I won't go on about that, but just to let you know it's going to be pretty serious. Now, this is just a summary slide, slide 9. I liked this slide, so I included it. It had a little bit more detail, and I thought it would flesh it out for anyone who's interested, especially if I always imagine that maybe somebody out there might want to look at my PDF and go over it a little bit more to try to get a better grasp of this than maybe just hearing me run it by them. This one shows amplifying hosts. Maintenance hosts is a good point. There are hosts that can carry it throughout the year, bats being an example, and dead-end hosts, and just these are vectors of this disease. Okay, let's see. This is from a WHO fact sheet. The United States just quit the WHO, which was one of the dumbest things I ever heard of, but it's a wonderful organization, and they have a lot of good information they put out in service to humankind. Signs and symptoms of Japanese encephalitis virus. Most of the time, it's asymptomatic, actually. If they do get infection, they can be mild, somebody feeling a little punk and no apparent symptoms, and then it blows by. But about 1 in 250 infections results in severe clinical illness. The incubation can be 4 days. Remember COVID, you had to be careful because people were contagious during those few days before they became symptomatic. So they were shedding virus and it was infective to other people. But 4 to 14 days, the illness will emerge. In children, and it's mostly children that are affected, gastrointestinal pain and vomiting might be the dominant initial symptoms. Severe disease with it can be characterized by rapid onset of high fever, headache, neck stiffness. Now, neck stiffness, the epistenus, where you can't tolerate stretch on your meninges, you try to tilt your head forward and touch your chin to, if I saw somebody in my practice that I thought could be developing meningitis or anything that inflamed the meninges, there's 3 layers of meninges. The thing I would have them do in an office setting is just touch their chin to their sternum or the front of their chest if they could. Bend their head way down. If they can do that, they're not doing too badly with their meninges. I saw a picture when I was in medical school of was a child in Brazil with meningitis, and his head was way back, his whole body was arched with a severe kyphosis, as though if you lay on your back on the floor and touched your head and touched your heels to the floor and had your body up in the air, like being possessed in the movies. That was the severity of pain the kid must have been feeling. I would think somebody with that much in the way of symptoms probably didn't make it. But at any rate, neck stiffness is something I want to emphasize. If you see that happening in somebody and that's new onset, don't screw around. You may have just a few hours to turn things around. And if you don't, they may die. It's that simple. Meningitis can take people out, and encephalitis can take people out in a very quick time. Disorientation, coma, seizures, spastic paralysis, and death are consequent of this. once the fire starts in the brain, it's hard to put out and hard to control damage. Fatality rate is as high as 30% among those with symptoms of Japanese encephalitis virus. 20 to 30% of survivors of the blatant disease have permanent cognitive, behavioral, and neurological sequelae, such as seizures, hearing or vision loss, speech, language, memory, communication problems, weakness of the limbs. There is no cure for the disease once it hits. It's mostly supportive care, and I might have done that. If you want to work it up, you need to do a lumbar tap, a spinal tap, and test the CSF, especially for IgM antibodies, which are the earliest antibodies generally in an acute infection. Treatment is basically supportive and can be palliative. You can't. There's nothing you can give really to try to fix it. You can try antiviral agents, but there's nothing that is considered a standard of care cure. Safe and effective vaccines are available to prevent Japanese encephalitis. And the WHO recommends that the JE vaccination be integrated into national immunization schedules in all areas where it is recognized. It's a public health issue. And it's horrific. Something that can be prevented isn't prevented. It's just unconscionable to me. So I think this is slide 11. This just shows the Japanese encephalitis virus structure. All the viruses I'm going to talk about today, there are 3 of them, are enveloped. There's a lipid envelope. And if you remember COVID and its spikes, this has spike proteins or glycoproteins that help it adhere to its targets. I don't want to get too bogged down with this, but this is a positive sense. In other words, the genome, it's linear and it's a long linear ribonucleic polymer, and it can be read just like messenger RNA from the human cell or from a mammalian cell. So it doesn't need the packaging like this. You'll maybe understand this better if you don't know about it. The group 5 viruses are negative sense, and they need to, their genome has to be protected from host cell defenses. So at any rate, there are about 180 copies of membrane proteins on the outer capsule of this, and the genome is in the center, of course. Now, that's about all I wanted to say about Japanese encephalitis virus, because I wanted you to be aware of it. So, next stop is Hendrix, Queensland, Australia. And there is a kangaroo looking for you. And if you'll notice here, Queensland has a projection up there along the coast that's just dipping its toe into the distribution of the Japanese encephalitis virus distribution. I'm sorry. So what's Hendra like? I think it's important to, if you talk about epidemiology, you need to understand the culture and the lifestyle of the place you're talking about. It's part of the job. I was not an epidemiologist, but I always was fascinated by it. I read about it a lot when I was a kid, starting when I was about 11, and was fascinated and deep admiration of Louis Pasteur and others in that history. But at any rate, in this, I just showed some nice pictures, which include, let's see, I gotta, Tom's Confectionery. I threw that in just to make you hungry. And there's a Dandelion Driftwood Cafe, just to show. There's a Brass Cafe. There's a lot of neat places to go and sit and enjoy yourself socially and with a coffee or tea. And middle to left to right, there's a home and a residential street in Hendra. And Hendra comes from a Cornish word meaning hamlet. And Queen Street Mall with a music performance is in the center there. And then bottom left to right is TC Bayern Park. And there's a couple strolling along looking happy. And horse races. Who among us has not, shed a few dollars, a few euros or some currency in horse racing? So horses are something we want to focus on a bit. In September 1994, mysterious respiratory disease, underlying respiratory disease, tore through a racing stable in Hendra, which is a suburb of Brisbane. Brisbane's right on that, I guess they call it the Gold Coast. It's just out at sea to the north. East is the Barrier Reef. And Australia is a fascinating place. When I was 15, I wrote to the Australian embassy and asked for forms and information to immigrate to Australia. I wanted to leave the United States already. And probably would have been better for all. At any rate, this is an unfortunate story in a way. There was this outbreak of this disease that was pretty sudden, and it tore through the racing stable. I don't know if you know much about racing, but racehorses ain't cheap. They're cultivated and babied, and they have to put out when it comes to it, but they are very valuable and beloved, usually treated quite well, and the kind of nobility of horses. And 21 horses were suddenly sick, respiratory and neurological symptoms. 14 died. That's 2 out of 3 died. And there were 2 humans in this run that died. One was a stable hand, or that got sick. One was a stable hand who survived, and this horse trainer, Vic Rail. And he's in that picture right there. He died several days after he caught the same illness from the horse. And the index case of this illness was a pregnant mare named Drama Series. So I have in the note card a highly, highly, highly recommended video. You want to see what the people who experienced this and dealt with it, what they were like and what they have to say about it. It's not very long and involves an in-depth interview with a veterinarian who took care of these horses and remembered it, and talked about how basically it was just a chamber of horrors. The horses in their neurological stupor and misery, and got to where they couldn't breathe because their lungs would fill with fluid, as did Vicks, I suppose. And they would smash their heads against the concrete and make open cuts, and just a nightmare. So they don't. It's not a gory video, but it's just for your understanding of what's out there. And one of the points I wanted to achieve in this presentation maybe is to save some of your lives, to emphasize that, you're not really all that safe. You think you are. And civilization, you got your house and your food in cans or plastic or whatever it is, and we're still children of nature, and there's no escape from that. And so there are things in the world that you better take seriously. So this led to discovery of the Hendra virus, and this was the primary reservoir right here. Australian fruit bats. There are 4 mainland species of Australian fruit bats, or flying foxes as they're called. They're huge. They like to eat fruit. Theropus, I guess, is the genus. I'm probably mispronouncing it. But they're the natural reservoir for Hendra virus. Hendra virus is abbreviated HEV. And again, named for the town. So hopefully you'll be able to remember it from these connections. These bats can carry this without illness, occasionally shed it in urine, saliva, and birth products. So let's go to the next slide. They carry it asymptomatically, and there's spillover to horses that occurs when horses ingest any kind of matter that might be contaminated with bat guano, urine, or birthing fluids. And Australian veterinarians noted from use of like these night cameras that are used on trails that bats roosted in the trees at night. They show the trees in this video that I saw, and you can see them shaking around as the bats climb around in the weaker branches, and when they come in to rest. And so they're shedding, doing their thing, even though they may be resting there. And so there's a concentration of their droppings and their secretions and such, bat guano and such, which can be contaminated with Hendra virus right under the tree. And this cam showed that the horses like to go under the trees and graze at night. And so one of the things they came to realize that, just making it so the horses don't end up having to having access to those more contaminated areas can help. Loss of habitat due to development, cutting of trees, wildfires, those kinds of things, roads. Bats end up drawn in closer to human circles to forage for food, because they got to eat too. They've been here at least as long as we have. And so all this can increase exposure to horses and other farm animals. The horses suffer greatly, unable to breathe, and it was hard on the vets to deal with it. But it came to one of the great success stories in veterinary medicine. They vaccinated the bridge host, which is the horse, and effectively broke the chain of transmission to humans. And it's more effective than they still encourage simple biosecurity measures like moving horse feeders away from flowering trees where the bats hang out. They literally hang out. And there's also an option of being able to use monoclonal antibodies. I'll talk a bit more about that sort of thing in a bit, but especially as post-exposure prophylaxis for humans. Now, 4 people, that doesn't seem like a lot, but people that work with horses are the ones at risk. And this was in terms of the extent of the spread of the disease, it was pretty focal and it was like a shot over the bow. But to my thinking, people that work in agriculture, and including with animals, and people like veterinarians, they expose themselves to a lot of risks. And I just wanted to take a moment to acknowledge that 2 of the 4 deaths out of 7 humans that had confirmed infections, so that's quite a high mortality rate, 4 out of 7. Dr. Ben Kunin and Dr. Allison Rogers, who were serving. It reminded me of all creatures, great and small. So now we're on slide 17. I have 51 slides, but a lot of them are going to be quick. So slide 17, no, I'm sorry. I'm on slide 18. I'll just say that from the pictures of those veterinarians who lost their life from this disease, catching it from the horses they were treating, it clearly doesn't matter how young and healthy you are, certainly it doesn't matter how tough you think you are. Nature don't care. So I would add that Hendra is not just an Australian anomaly. It's a blueprint for understanding how environmental degradation drives emerging infectious diseases globally. So let's see. I seem to be. Yeah, here's the slide I want. Alpacas are very interesting in this. They found that they can, in collaboration with Chilean sources, they can inoculate alpacas with inactive viral components, and they'll develop very small, heat-stable, what they call nanobodies, which are DS90 is the term for it, and they bind to the surface protein and prevent the Hendra virus from entering human cells. And so I wanted to acknowledge these University of Queensland, Dr. Ariel Isaacs and Professor Daniel Watterson and their support staff were instrumental in developing this, and it's a potential therapeutic treatment to prevent HEV or Hendra virus. These antibodies, interestingly, are about 1/10 the size of conventional antibodies. So they can really get in little crevices and little tight spots on the surface of which is very convoluted and irregular of the and has the envelope also that protects it of the Hendra virus, and knock off the effective glycoprotein that brings about adherence to cells that human antibodies can't quite do so easily. So let's see. Now, this is slide 19. And just here, I wanted to say horses, once they're infected, can spread the virus to people, and they tend to get sick real quickly. I want to talk in a minute about why. That people get the Hendra virus after contact with tissues. It's a bit of a summary. CDC had this, and I wanted to show. I don't think it's of the quality, this slide, of the information that I got from WHO, which I used to supplement some of the articles I read. But one of the points I wanted to make is, to date, there's been no spread between people or direct infection from flying foxes to people that have been documented. But if someone has a contact with an infected horse, they better have personal protective equipment, boots, gloves, protective eyewear. You get secretions in your eye. You can have the virus introduced into your tears that drain through the lacrimal ducts to your nasal cavity, and then eventually get swallowed. And so that'd be a bad way to lose. Masks or respirators and gowns are the ticket. And so I kept the same slide. I like the flying fox here. This is just to mention that there's no human vaccine for Hendra virus at this point, but some antiviral drugs have been tried, and I won't belabor that. I just wanted to mention it. So that's the story of Hendra that I'm going to tell today. And now we go to the focus. Next stop, the village of Sungai Nipah, near Ipoh in the state of Perak, in West Malaysia. Malaysia is an amazing place in a lot of ways. For one thing, geographically fascinating. It's a peninsula at the tip of an isthmus there from Thailand, but also, and that's West Malaysia, and East Malaysia is across a sea. There's open ocean between two parts of the country, and East Malaysia is on the northwest coast there of this neighboring island. And I wanted to make a couple points about this. You know, you hear about countries that, have an epidemic or something. You think, oh, well, there's like, I think of the current American president talks about other countries that he doesn't know anything about with disdain and arrogance. And people that haven't been to Malaysia might like to think it's a third world nation, because, if you live in modernity and you think you're better than everyone else, then that works for you, I guess. But I would point out Malaysia has a high adult literacy rate of about 95%. It's ranked first in Asia and 24th globally for English proficiency. They probably speak English better than most Americans. I remember the English guy who told me he thought he spoke English until he came to America. I'm hearing about space elevators. Is there someone talking? It's number one globally in preferred medical tourism. It's for people starting a business. It's about the 7th best place globally. And education is 37th overall, which is better than the United States. It's for retirement, the 27th most comfortable destination. And there's a lot of other good features about it. But I wanted to point out that it's really quite a place. And I've got a series of slides here to just, this was what I had intended to have on. This is Ipoh in the Kenta district. It's the capital city of Perak, Malaysia. Perak is where that's sort of on the eastern side of the peninsula of West Malaysia. That's Perak is where this Nipah virus came from. So, few slides just to show you pictures. We'll go quickly through these. They have looks to me like a great agricultural place. They have sweet corn, shallots, just a couple of examples of the kinds of vegetable products and agricultural products they grow. These are rice paddies, huge and well organized. And, you know, it's there's kind of a connection, as I understand it, between growing rice and Zen. If you are mindful and do the right things, you can have great output. It's very effort related. And they also have horses. And they have horse races. So they have a lot of chicken farming, and some of the chicken farms are very progressive, as is this one, which has more free ranging. And chicken is their favorite meat. But this is a butcher carving up pork. And pork has an interesting history. It was initially subsistence farms, small operations by Chinese, ethnic Chinese that lived in Malaysia that grew and pigs and part of what they lived off of. And in the 70s, it became more and more popular and grew. And as of 2023, there were 401 pig farms in peninsular Malaysia and 118 in East Malaysia. So that's pretty good for that size country. But that's indicative of some recovery, because the pig farming industry took a big hit, which I'm going to tell you about. And these are these sentient creatures who feel fear and would like to live are some of the pigs that are being raised for humans' consumption. This is the town of Nipah. It was quarantined, I guess, 1999 by the Malaysian government because they had this outbreak of a disease. And some of you may remember the movie from 2011 with a star-studded cast called Contagion. Contagion involved a pandemic that burned through society, and I won't go into details about it, but this is just a collage of pictures from that movie. That was inspired by what happened in Nipah, if you didn't know. And it might be interesting to go back and watch that movie again with the understanding of what you learned from this presentation, or I hope that you learn. Let's see. Give me 1 second here. This is from the Telegraph, the UK newspaper. They have Asian Bangkok offices and cover Asia directly. And initially, the Malaysian government, when this thing broke out, and suddenly people got sick, and I'm going to tell you kind of the course of that. They thought it was from mosquitoes. So the Malaysian government was trying to do major spraying of and treatment of waterways and all this sort of thing. It's a tropical sort of place, humid and not far from the equator. And it was learned by someone who I'm going to tell you about that the pigs were infected with this disease, and the pigs were rounded up, and they shot them till they ran out of bullets. And then they used wooden sticks to, quote, destroy the pigs, unquote. And then the corpses were scooped up by diggers and unceremoniously dumped into deep, muddy pits. And in reading I did, people that had experienced this, they said the villages went empty. The smell was horrific, and the farms were empty and desolate. And so I introduce to you a modern hero, and this is Dr. Chu Ka Bing. And I would say his tagline, as I like taglines, could be stubbornness can be a sign of high intelligence. I'd like to tell you the story of this. First off, imagine he was a graduate student in medical microbiology at the University of Malaysia in Kuala Lumpur. And the outbreak began in 1998, and it was misidentified as, guess what? Japanese encephalitis virus, because that's what they were so used to seeing. So they thought this must be that. Now, he looked at samples of it and found them alarming, but let me tell you more about that in a minute. I'll tell you how the whole thing started. Just imagine it's a humid night in a small village in the Kentha district of Perak in Malaysia, late 1998. A 29-year-old pig farmer, we'll just give him the name Amir, arrives at a rural clinic with what looks like a bad flu. He had fever, pounding headache, exhaustion. Not that unusual for the season. But by morning, things were really going south. He was confused. He couldn't remember where he is. His speech is slurred. His wife says he was perfectly fine 2 days ago. So the clinicians suspect Japanese encephalitis. That's the usual culprit. They began supportive care. The next day, Amir was in a coma. And he's not the only one. Across the region, farmers, veterinarians, and slaughterhouse workers were arriving with the same terrifying pattern: flu-like symptoms, rapid neurological decline to coma. The usual tests came back negative. Treatments didn't work, hospitals filled up, entire farms were shut down. Thousands of pigs were culled. No one knows what's happening. In the end, it was over 1 million pigs that were slaughtered. In March 1999, a team of scientists isolates a virus no one has ever seen before, a virus capable of jumping from bats to pigs to humans, causing severe encephalitis with a fatality rate that was extraordinary compared to most pathogens, on the order of 70-75%. And they named it for the village, Nipah virus. The world realized that something new had come to our doorstep, a deadly multi-species epidemic. The first isolation of it was achieved by Dr. Chu Ka Bing, who was then this medical microbiology graduate student. And it began, he noted it was misidentified as a Japanese encephalitis. He looked at samples under a microscope, and he was alarmed. I really admire this. If you ever worked with microscopes, I got a number of microscopes. I got medical quality microscopes that I can use to look at things. I think it's a wonderful thing to be able to do, and I think it's maybe a mistake when people get too far removed from traditional technology like that. But it's an important tool for observation. Well, he looked in these tissues that were supposedly from patients or animals that had, patients, in this case, lungs of Japanese encephalitis. But he saw syncytia. Syncytia means that the cells melded together. They blended together into giant cells, multinucleated giant cells. They weren't separate cells anymore. That's unusual. And he actually, especially in the lung and brain tissue, he found this. And he recognized these distinct cellular abnormalities. And based on those, he felt that this was a paramyxovirus. It's a hallmark of that kind of virus. And one of the types of that was a syncytial virus. And also the rapid destruction with widespread tissue necrosis. That was a really aggressive pattern. So he showed it to his advisor, his graduate advisor, who dismissed it and said, don't forget about it, destroy the samples, that's contaminated. He just didn't think out of the box. And Dr. Bing, not a doctor yet, but he was on his way, he felt sure this was something different, and he wouldn't give up. He persisted, as some would say. He wrapped up his samples, smuggled them, in a sense, into the United States and brought them to the CDC in a desperate attempt to identify this mystery killer that was being misidentified. He flew to the United States with live virus samples in his suitcase and took them to CDC in Fort Collins, Colorado, and Atlanta. And though he broke standard safety and quarantine procedures, it resulted in the realization that this was not Japanese encephalitis, but a new virus. And it was not related to mosquito-borne diseases, but rather to the pigs. And so it led the efforts of the Malaysian government to change to culling the herds of pigs, which was a huge agricultural and economic hit, but it was a mode of survival, really. So his own research showed the virus was much larger than Japanese encephalitis and did not respond to the mosquito control efforts. So at any rate, he saved countless lives. And his story was a key inspiration for this movie Contagion, and I'll let you maybe go back and watch the movie and see if you can figure out what character they had that was closest to his courage. And he's won awards, and he is considered a hero in Malaysia. My hat's off to him. So this is just a map of the distribution. I need to really step it up here. And it's in Bangladesh and southwest India. Although the fruit bats are all across Southeast Asia, it's been interesting that they haven't seen more of this more widely. But at any rate, well, this one's in with that actually would apply more to the Hendra virus, that comment. But let's see. So mortality can be very high. I just lost view of my screen. Sorry. 80% in Bangladesh when it hit there. That was a different strain. There are two different strains that are known. And Philippines, they had contact with horses Kerala, India got recurrent outbreaks, and they pretty much, Bangladesh pretty much gets it yearly. The United States has seen this as a Category 3, a Category C bioterrorism threat, as it could be, quote, engineered for mass dissemination in the future, which tells me the United States probably has a stock of it. So let's see here. This is a diagram. I just liked it because it showed the vectors and how this virus can be spread. And I'm going to make some jumps here. I encourage you to look and hopefully enjoy my PDF. There are 6 proteins involved in this, and on the surface, and its envelope is derived either from endoplasmic reticulum or from cell membrane, probably endoplasmic reticulum, because the mRNA fragments that the virus creates for translating to G glycoprotein, which is the one that it's like the head of the arrow that attaches to the receptors, and the fusion protein. Those are in the rough endoplasmic reticulum, and our ribosomes bound to the endoplasmic reticulum, and so it's thought that that's related to them being bedded out of the cell and involved in the final assembly of the virus. It has a lipid bilayer, that's this envelope that's probably host-derived, a matrix protein, and then viral RNA, and its packaging means it has these 3 nucleoprotein polymerase, a large polymerase, which is L for large, and phosphoprotein, that are in association with this long negative sense strand of RNA. And when it gets excreted, the N protein protects it from nucleases in the host cell that would eat it alive. And so the first thing it sets about doing is transcribing little messenger RNA segments. And so here's the thing. There's the receptors in humans are ephrin B2 and B3. They are highly preserved in mammals, and which explains why there's all these different species that can get this. And also the endothelium, or lining of the blood vessels, which is your transportation system for nutrients, oxygen, everything that keeps you alive in your body, are loaded with these receptors. And so that's a primary target for this virus, for that glycoprotein, the G protein on the surface of the virus. The other is that the brain, human brain, is loaded with ephrin B2 and B3 receptors. And so this is not just vasotropic, but neurotropic, and getting like an acute respiratory distress syndrome, ARDS, which is horrific. I hated it when I saw patients get that, because it meant that they ended up with them one more organ system out. They were so likely to die. 2 organ systems out, and you're really up a creek. But anyway, compromised lung function, suffocating, even with ventilator support, can happen. But when it gets into the brain, because it's neurotropic, and because it's in the blood vessels and it inflames them, the syncytial, the F protein on the surface causes the cells to meld and make giant cells, and that causes massive inflammation. So you get vasculitis everywhere, and it damages the blood-brain barrier, and it gets into the central nervous system, and then it can really do you in. And an alternative route is via the olfactory nerve. It's the old thing to keep your nose clean, because the olfactory nerve is a direct route to your brain for pathogens. So if it gets into that, it can easily spread through the ventral cortex of your brain, which is, we used to say, getting up there where you live. I used to do operations on sinuses, and it went up right to the skull base and anterior cranial fossa, and you didn't want to go through there. That has happened. Not by me, but I mean, it has been known to happen. See, I'm trying to get this to, hold on, I'm having trouble progressing my slide. Okay. There's a newspaper article described how they took live pigs and forced them into pits. It kind of breaks my heart seeing this. I don't know at you. These are sentient creatures, like I said. This is a farmer, I remember his name, who lost his farm. They had 3-generation farms that ended up empty because of Thomas Wong. He's 63, pretty youthful looking, and his farm is done. His livelihood gone from this. This is an abandoned farm from this disease. This sort of disease impacts agriculture, veterinary medicine, and economics, and human medicine. So this is just description of the things that happens when the virus does its thing. And that's more the molecular biology sorts of aspects, or the virology aspects of the mechanisms. I'll let you go through it. Interestingly, this versatile LP complex can transcribe shorter strips of messenger RNA that are ribosome ready for translation, and whereas the negative strand RNA, and when it makes a positive strand as a template, that's uncapped and not polyadenylated, adenylated. So it's got to be protected because it's strange to the cell. And the bottom line, the vasotropic knife that this Nipah and Hendra virus can enjoy causes rip-roaring vasculitis. It can fill the lungs with fluid, causing hypoxia, which causes confusion and death. And brain fever is an old term that goes back to Dostoevsky, encephalitis. And here's the structure of the Nipah virus, which is pretty much what I've been talking about. The segments, the 6 segments there at the bottom are of the proteins are important. One last thing I'd like to mention, I'm almost done, and this is really cool, is that they can't safely work with this live virus, but if they take pieces of it, they can make kind of a mini assembly that they can test and have replication, and they can insert something in it to see if it's working. And when they did that, what they found is if they had in their assembly, many viruses can cause encephalitis, but that's a little bit more than what I can do here. But here is the really cool thing. when the number of ribonucleotides are not a product of 6, then it didn't work. And I point out here, just a summary slide, Malaysian strain has 18,246 bases, and that's 3,041 times 6. The Bangladesh strain of Nvidia, that's a company, Nipah virus, has 18,252 bases, which is a multiple of 6. And the Hendrix virus, similarly, they had the same kind of equipment. They're cousins, and it has a multiple of 6 bases. And the reason, what they found was the LP polymerase complex grabs onto 6 nucleotides at a time as it works. And if it's not 6, it comes to a rest. It comes to a halt. And that gives a strategy for potential treatments. Currently, there's no vaccination for Hendrix virus or Nipah virus for humans, but they do have a Hendrix virus vaccination and a vaccination for horses, and a Nipah virus vaccination for pigs. And one of the reasons I wanted to kind of indulge in the environment of Malaysia in this, aside from the fact that I think it's a beautiful country, is that if it can happen in Malaysia, it can happen to your country. It can happen anywhere in some form. So be prepared for zoonoses jumping to humans. And don't vote for idiots that want to disassemble everything that works in taking care of this stuff. And my last is to honor Dr. Chua Ka Bing, who had the guts and chutzpah to take live viral samples to America. And I couldn't help but think that these days, if he did that, when he got to Atlanta, that ICE would show up and beat him up and throw him into a concentration camp. But this was back when there were different practices, I guess, different attitudes. So if you have something like this, medical emergencies and epidemics, and who are you going to call? You're going to call Dr. Chua Ka Bing, or someone like him, maybe like Steve Lesnar. Or if it's a chemical spill, Mike Shaw. You know, we have a lot of expertise here. And that is it. I stuck this in front of my beryllium talk, and I appreciate you. I tried to keep under my time, and I hope it made sense. Any questions? I think there were questions that, like I'd said, I was trying to keep my eye on the clock, and on my notes I would stay on track, and I didn't watch the Dolly needs